Hello, my name is Udita Rathod. I am going to present on the topic of isolation of bacteria by various methods. There are many ways to isolate bacteria. One of the method is spread plate method. So, here's the animation of spread plate method. Firstly, we have to take sterile agar mutant agar plate, sterile cultural tube and sterile pipette. The first step is to inoculate the plate. Pipette a small fixed volume of the diluted sample like zero point 0.1 ml on the center of the nutrient agar surface. Then spread the sample. Carefully spread the culture evenly across the entire agar surface using the sterilized spreader. Rotate the plate to ensure complete coverage. Now, after spreading, incubate the plate. Allow the sample to absorb into the agar. Then invert the plate and incubate it at the appropriate temperature. Temperature is 37 degrees Celsius for 24 hours for microbial growth. Then count and calculate. After incubation, count the number of visible colonies on the plate, which represent the colony forming units. Aim is to enumerate the number of viable microorganisms per milliliter in a liquid sample. Principle: when a diluted sample is spread on agar, each living microbes grow into one colony. By counting these colonies, we can find how many viable microorganisms were in the original sample. The formula for counting it is the colony forming unit per ml is equal to number of colonies by volume of cultural plate in ml into total dilution factor. The requirements are the sterile nutrient agar plate, sterile cultural tube, sterile pipette, sterile spreader, sterile petri plate and incubator. Now procedure. Firstly, we have to prepare serial dilutions of the sample by sterile water or saline water. Then inoculate the plate of 0.1 ml of a specific dilution on the center. Then spread the sample gently and evenly across the entire surface of nutrient agar plate. Then incubate the plate for few minutes at optimal temperature for the microorganisms. Then count the colonies after incubation. Count the colonies from the plate. Multiply this number by the dilution factor to get the original concentration of the sample. The result would be the formation of evenly distributed, discrete colonies on the surface of an agar plate after incubation, allowing for the isolation and counting of viable microorganisms. The number of colonies is used to calculate the colony forming units per milliliter in the original liquid sample, providing a measure of microbial density. Thank you.